Okay, let's continue our story on glucose catabolism, and we're going to talk more about what happens during cellular respiration. Everybody's favorite topic in biology. I know you're excited. Let's talk a little bit further about it. And again, don't get wrapped up and scared by all the terminology here. It's basically, we're really just kind of taking a fairly broad overview of what the purpose is and how this energy is moving out of glucose molecules and being temporarily stored in ATP fuel molecules. That's the whole key point here, and we want to become familiar with some of the processes that cells can use to achieve that goal. So in the last video lecture, we introduced that cells can use respiration or cellular respiration as a process for oxidizing glucose, breaking it down, and harnessing energy stored in the bonds of glucose to make ATP. And then fermentation is an alternative process that some organisms can use. And both of those processes follow glycolysis. So glycolysis, the partial oxidation of glucose and conversion of that into 2-pyruvic acid molecules, is a common phase to both cellular respiration and fermentation. All right, so now let's focus in on what's going on during cellular respiration. Cellular respiration is a very complex process, and we're not going to get into the nitty-gritty, gory details of what's going on. We're going to kind of stick with a broader perspective on it. Okay, so let's take a look at what's going on our diagram over here. So for every glucose molecule that goes into glycolysis, you wind up with 2 of these guys, pyruvic acids or pyruvates. And then those are going to feed into a multi-stepped process over here called the Krebs cycle or the citric acid cycle. And your book has more details on that. Use your study guide for guidelines on what you need to know. But that is a series of chemical reactions, step by step, and it continues the oxidation of glucose. And when the Krebs cycle completes, for every molecule of glucose that you started off with up here, that molecule of glucose has been oxidized to 6 carbon dioxide molecules. So glucose has 6 carbons in it. Those 6 carbons are now in these 6 carbon dioxide gas molecules. So this diagram over here is showing you CO2 as an output of the citric acid cycle or the Krebs cycle. And you can look in your textbook for more details of what's going on during the Krebs or citric acid cycle. Many chemical reactions, they take place in a cycle type form. We're not going to go into all of the details on that. And a major output will be these carbon dioxide molecules. So have you ever wondered where the carbon dioxide that you exhale out of your body is coming from? It's coming from the oxidation of your organic nutrients that you've absorbed from the foods that you've eaten, and those have been taken into your cells and they've been oxidized, and those carbons have wound up in CO2 molecules. And CO2 cannot accumulate in your body because it causes your body fluids to become acidic, so you've got to exhale it out. If you haven't studied that yet, you will in your Biology 202 class. And same thing for microorganisms. They're little single-celled microorganisms generate carbon dioxide. Carbon dioxide can diffuse through the cell membrane, so it diffuses out and heads off back into the atmosphere. So, when we hear about carbon dioxide in our atmosphere, a lot of that is coming from the living things on this planet performing cellular respiration and giving carbon dioxide off back into the atmosphere. Okay, and notice another output over here, ATP. For every glucose that enters cellular respiration at the beginning, goes through glycolysis, the cell gets 2 more ATPs. When you perform the Krebs cycle, you also get 2 new ATPs for every glucose molecule that starts. Now, if you go into the detailed process of the Krebs cycle, you actually run the Krebs cycle twice for every glucose that you start off with. And that's because you've got 2 of these pyruvic acids for every glucose. Each of those feeds independently into the Krebs cycle. So you run the Krebs cycle twice. Every time you run the Krebs cycle, the cell gains one new ATP. All right, so after running the Krebs cycle, that still sounds kind of wimpy, right? We started off with glucose, and we've gone through all this rigmarole. We went through glycolysis, we went through the Krebs cycle, and the cell has only gained 4 new ATPs at this point. That sounds pretty lame. And it is pretty lame for the most part. You've gone through all that effort, and you've only got 4 new ATPs to show for it. And we've already said that cells that do cellular respiration are going to make lots of ATP from breaking down glucose. So the majority of that ATP gets made during the electron transport chain. And so we'll talk a little bit more detail about what's going on there. The electron transport chain is going to involve these electron carriers, like these NADHs that you see here. Everywhere you see one of those get generated from the oxidation of the glucose. Hydrogens are getting plucked off of the glucose and its breakdown products as it's being catabolized, broken down, and transferred onto NAD+, and those become NADHs. And we call those electron carriers because they're temporarily carrying or hanging on to those hydrogens and electrons that were originally on the glucose, and they're actually going to donate those into this multi-stepped process we call the electron transport chain. Another thing I want you guys to notice on this diagram over here right over in here. Notice there's an O2 oxygen gas input on our electron transport phase over there. Oxygen gas is actually used at the very last step of the electron transport chain, and oxygen gas molecules get converted into water molecules, as we'll see as we talk in a little bit more detail about that story. So that's why you need, if you are an aerobic type of organism like we are and many microorganisms are, the reason you must have oxygen gas is because it is used for the very last chemical reaction of the electron transport chain. All right, so something else I want to point out over here. If you are an aerobic organism, these processes require O2 if you're an aerobic organism. So we sometimes call these aerobic processes. And the reason for that is if for some reason you're an aerobic organism and you don't have access to oxygen gas, your electron transport chain shuts down, and so does your Krebs cycle. It's kind of like a toilet backing up or a sink backing up. If oxygen gas is not available for the last step of those phases, then the step before it can't happen. The step before it can't happen. The step before it can't happen, and it shuts down all of those processes. Okay, on the other hand, glycolysis and fermentation are called anaerobic processes. Anaerobic means without oxygen gas. Oxygen gas is not required. You don't need oxygen gas to do glycolysis, and then fermentation can follow glycolysis. So keep in mind that those processes are anaerobic. They don't require oxygen gas. And you may have covered some of that in your anatomy and physiology class as well. Usually you cover it when you're thinking about skeletal muscle fibers and how some of those can shift over on a temporary basis and do fermentation to make their ATP, and they generate lactic acid as a waste product of those processes. That's only something, and we can't do that on a long-term basis. It's very, very temporary because you just don't make enough ATP for survival if you're trying to rely on fermentation. Now we'll see some microorganisms, they can get by just doing fermentation. They don't have to do all of the cellular respiration rigmarole to make all of their ATP. All right, so let's take a look at a summary So if we do aerobic respiration, so all of this, starting from one single molecule of glucose, what do we get out of that? Okay, so we've already talked about how we have three major phases over here. The first one is glycolysis, and we talked about how for every glucose you're going to make 4 new ATPs, but you use 2 to get the processes started, so it's a net gain of 2. Synthesis of acetyl-CoA, I didn't really go into that, but that's a step that precedes the Krebs cycle. So when you do that, plus the Krebs cycle, 2 new ATPs made for every glucose that you start off with. And then in prokaryotes, and we're mainly thinking about bacteria here, in many or most prokaryotes that do aerobic respiration, so this is when oxygen gas is involved, running the electron transport chain, this last phase over here, you get 34 new ATPs for every glucose you started off with. So that's where you're getting the most bang for your buck. You total them all up, the net gain for the cell in a bacterial cell can be 38 new ATPs. In a human cell, it's actually going to be a little bit lower. When you guys study cellular respiration and anatomy and physiology, or another biology course, you no doubt learn that the mitochondria are the sites where most ATP is generated. They're the powerhouses of the cells, and that's because that's where the Krebs cycle and the electron transport chain takes place. But they actually don't make quite as many ATPs per glucose as a bacterial cell will do, because it requires some energy use to get the pyruvic acids from glycolysis, to get those converted into acetyl-CoA and sent into the mitochondria. That actually uses up some energy. So the cellular respiration is actually a little bit less efficient in a eukaryotic cell like we have in general. Now on this table over here, notice it is also telling you the number of NADHs produced. NAD plus becomes NADH, and the hydrogen and extra electron that make that happen are coming off of the glucose, and they're temporarily storing those. And then there's another electron carrier called FAD, which essentially does the same thing. It's used over here instead of NAD plus at a step during the Krebs cycle, so you make those as well. Same kind of thing. It's an electron carrier temporarily hanging on to a hydrogen and extra electron from the glucose. NAD plus and FAD are both vitamin derivatives. So that's one of the reasons why you need to make sure you take your vitamins. You've got to have the proper vitamins so that things like NAD plus and FAD are available for your cells to run cellular respiration. And it's the same thing for other living things as well. All right, so that's the overall summary and the different phases of cellular respiration and where all this ATP is being made. Let's talk a little bit more about this mysterious electron transport chain, though. What is that? What does that mean? It sounds like a bunch of gobbledygook. So I've been talking a little bit about these NADHs and also this FADH2 molecule that acquired a hydrogen and an extra electron from glucose. And then I said they're going to temporarily hang on to those. And what they do is they donate the hydrogen and electrons into the electron transport chain. And what is the electron transport chain? The electron transport chain is a series of chemical reactions. They're oxidation reduction reactions where NADH and FADH2 or FADH2 give up those electrons they acquired from glucose to another molecule. And then that molecule hands them off to the next, and the next, and the next, and the next, on down the chain. That's why it's called the electron transport chain. So as one molecule becomes oxidized, if NADH gives up electrons, it becomes oxidized. This next molecule becomes reduced. and then that FMN becomes oxidized as it gives off electrons to that molecule called coenzyme Q. And then that one's going to become oxidized as this one becomes reduced. Oxidized, reduced, oxidized, reduced, and so forth and so on. So that's called the electron transport chain. And as that's happening, this flow of electrons from one molecule to the next, to the next, to the next. I know it's kind of hard, it's very abstract to think about, but there's energy associated with that flow of electrons. That flow has energy associated with it. That energy is captured and used to make more ATP. And it's a very efficient process. Down here at the end, there's good old O2. So if you are doing aerobic cellular respiration, oxygen gas is called the final electron acceptor. It's the final molecule that's going to undergo reduction and receive electrons from a previous molecule in the process. And as it does that, as O2 molecules receive electrons, they also combine with hydrogen ions. So in your cell fluids, you always have hydrogen ions floating around. If you guys have studied pH before, and hopefully you have, you know that pH is a measurement of the concentration of hydrogen ions in a fluid. So that just emphasizes how important they are. They're always going to be around. And as the oxygen gas molecules acquire electrons, hydrogen ions attach, and the oxygens get converted over into water molecules. And I know it can be kind of confusing that typically that is written as 1/2 of an O2 gets converted into a water. And to me that's kind of weird. What in the world are you talking about, 1/2 of an O2? To me it makes more sense to say that a single O2 gets converted into 2 water molecules as part of that process, instead of 1/2 of an O2 becoming a single water molecule. So remember when we went over the overall chemical reaction of cellular respiration, we talked about how oxygen gas is an input and water is an output. So aerobic organisms like us and many microorganisms actually generate some of their own water. That's called the water of metabolism. All right. Now, do all organisms that do cellular respiration and run an electron transport chain use oxygen gas at that last step? Absolutely not. Anaerobes, organisms that are capable of doing anaerobic respiration, substitute something else for the O2. So they don't require oxygen gas. Remember, we studied these kinds of creatures in Chapter 6. If you're an obligate anaerobe, actually, oxygen gas is toxic for you, so you've got to stay away from oxygen-rich environments. But if you're going to run cellular respiration, you've got to have something you can use as a substitute for oxygen gas at the end. And then whatever they're using as a substitute, it could be sulfate, nitrate, nitrous oxide, various things that different microorganisms are capable of using. Those will get converted into alternative products. So instead of generating water from metabolism, they'll generate some other type of product at the end instead of water. This diagram from your textbook is giving you another visual on the electron transport chain. Again, we're not going to go through all of the gory details. But when we look at that previous diagram and we see electrons are being handed off from one molecule to the next, to the next, to the next in the electron transport chain, where is that happening? How does that all stay coordinated? All right, and in bacterial cells, it occurs in the plasma membrane, the cell membrane of the bacteria. So here's your peptidoglycan cell wall. So this looks like a chunk of a gram-positive cell envelope. Remember that. There's your cell membrane. So all these these molecules that hand off and receive electrons, one to the next, to the next, to the next during the electron transport chain, they're actually embedded within the cell membrane. In eukaryotes like us, all of these molecules are embedded within the inner membrane of the mitochondria in the cell. Okay, so you have electrons that are being handed off from one molecule to the next, to the next, to the next, and then finally those electrons are sent over here to oxygen gas molecules, and then those oxygen gas molecules, as they become reduced, they bind with hydrogen ions and they're converted into water, H2O. All right, and then something else that happens during this process, there are hydrogen ions that are involved here as well. So we didn't just have electrons that went that bonded to the NAD pluses or the FADs. Hydrogens bonded as well, and those have now been converted into hydrogen ions in the electron transport chain. And so they're over here on this side of the cell membrane. As the electrons are flowing from one electron carrier to the next, there's energy associated with that. And that energy helps drag these hydrogen ions, if you look closely at the diagram, from one side of the cell membrane to another. Okay, it's a type of active transport. Remember how we talked about active transport before, where you are moving a substance across the cell membrane against its concentration gradient. And what happens is, over here on this side of the cell membrane, I know this looks like I'm scribbling like a 2-year-old, you have a buildup of hydrogen ions. Now I want to draw your attention over here to that molecule. That is a special protein, an enzyme called ATP synthase. And so guess what ATP synthase does? Hopefully you're thinking that it's going to synthesize ATP. So basically you wind up with a buildup in concentration of hydrogen ions on that side of the cell membrane. The ATP synthase has a channel in the middle of it that allows hydrogen ions to flow through. So we talked about how protein channels and membranes are sometimes very specific about what they allow to pass through. You learned about that in Biology 201 as well when you study the muscular system and the nervous system. So it's almost like you've got a waterfall of hydrogen ions flowing through these ATP synthase enzymes. So there's energy associated with that, just like you have energy associated with the flow of water across a Tennessee Valley Authority dam, and that energy can be harnessed to generate electricity. Same thing here. As these hydrogen ions flow through these ATP synthase enzymes, there's actually a propeller-like feature on here, and I know it's weird to think about when you're discussing protein molecules having features on them that look like propellers, but they do. And as the hydrogen ions flow through there, the propeller is turning, and ADPs and phosphates are captured and bonded together to make ATP. That's just kind of an interesting little aside there on how, why does this flow of electrons lead to the formation of so much ATP? And this is just a very efficient way of capturing. Energy changes forms, and this is a very efficient way of capturing and harnessing that energy to make as much ATP as possible. And then I do like that this diagram is showing you in aerobic respirers, organisms that require O2 for survival, they use the O2 as the last step of the electron transport chain and convert it over to water. Anaerobic respirers will use other types of molecules to receive the electrons at the last step and convert those over to products other than water. All right, so I know that's kind of a heavy-duty topic going through cellular respiration, and again, try to focus in on the bigger picture parts of what we've discussed as I've gone through that particular topic, and don't get too bogged down and lost in the details of your reading. If you are confused, be sure you, first of all, try to post some questions on the discussion board so we can go over things there for everybody to be able to see, and or contact me and let me know that you need some assistance. The next video lecture, we're going to talk more about this fermentation alternative. So we said cellular respiration and fermentation have glycolysis in common. And then the two pyruvic acids will either go through cellular respiration, or in some organisms, fermentation can be used as an alternative. So we'll talk more about that process. It winds up being really important in microbiology for identifying and diagnosing what types of organisms are causing infections. And then also lots of things that we use on a daily basis are made through fermentation by organisms like bacteria and yeast. So we'll talk about that as well.